Good morning, all of you. In the third session, we'll be discussing the concepts of protein structure prediction. All of you will be thinking what is the need of protein structure prediction. In the last session, we have been discussing regarding X-ray crystallography, NMR, cryo-electron microscopy, the terms. We were discussing that researchers have used these technologies for the determination of protein structure, means experimentally finding the protein structure. Okay, experimentally resolving the protein structure. We discussed the concept of Armstrong. So what is the need of protein structure? Okay, so but before we go it, I'll ask you a small question. The Ramsden score defined by the energy of protein structure, is it right or wrong? Or does an energetically stable structure have a higher Ramsden score? You'll say, oh yes, of course. Understand that this is just the favorable positioning of the diagonal angles on the map, okay, which is defined by the energy. Understand? So both the terms are like overlapping. Only when they are energetically favorable conformation, then only they localize the fitting conformation map in the fitting position in the map. Otherwise, they'll go somewhere else. Okay, so within a protein structure, anywhere, whenever a protein energy structure energy is not very low, okay, not the lowest, which is functionally active, that time the protein structure energy will be much higher. Okay, it will not be like the diagonal angles of the amino acids won't be fitting within the defined region of Ramchandran. Okay, so it will be going away, so the Ramchandran score goes down. Okay, but it is not a hard and fast rule, understand that. Because one protein structure can take multiple shapes. So even if it is possible that amino acids take up some diagonal angles which are like outliers in the outlier zone, which is not allowed by the Ramchandran. This is quite possible. So such regions are termed as, like such amino acids are termed as Ramchandran outliers. They are there, but still the protein is functioning. Understand the terms, okay? Alright, so but before we go further, we should discuss why there is a need of structure prediction. Okay, so if we carefully see here, I have taken this diagram from UniProt KB and Tremble database, means Universal Protein Knowledge Base and translated European Molecular Biology Laboratory, the complete database, including the translated DNA sequences also into the protein. So this has crossed 200 million in 2024. 200 million. See the huge number, substantially large number. On the other side, if you just see the SwissProt entry, means the UniProt KB knowledge, Universal Protein Knowledge Base, the SwissProt part, which are the manually annotated sequence entries, they have also crossed 600,000. Okay, and compared to this huge number, what is the structure of a protein? Like as we have been doing solving the protein structures from a long time. Okay, so this has just crossed 2 lakh entries recently. Only 2 lakh. See, compared to 6 lakh entries to here 2 lakh entries in SwissProt, and 200 million to 6 lakh. If my sequence is a new sequence, it's a completely unexplored sequence and I do not do share with anybody, then I need to do everything myself. In that case, if I first sequence the gene, I find that it is not similar to any similar sequence present in the database. Okay, then what should I do? Here if I just see it is 200 million, I can dump it just another one more sequence here in 2200 million. Okay, I don't have manually annotation, I need to do everything myself. And if I want to solve the structure, it takes another methodology, another problem. The reason is the limitations of structure determination methodologies. Means protein structure determination methodologies are not sustainable enough to do it very fast to match the scale. Here the rate is much more high, but here the rate is much lesser, and this gap is constantly getting increased, and we are unable to bridge the gap between the number of sequences and the number of structures. Number of sequences are going very fast. Okay, every year number of sequences are getting increased substantially faster. On the other side, the number of structures are increasing very slowly every year. So you understand that there are certain limitations in the protein structure determination methods. Okay, so what are the limitations? You see, the limitations are X-ray structure when we are doing. Okay, the machine itself is very costly, which you don't find anywhere. Still if I do it, still if I am able to manage the machine, everything done. Then the biggest trouble comes is my protein should be a crystal, 100% protein crystal. So the trouble comes is many proteins do not crystallize at all. Okay, they don't form crystal. Okay, so what researchers or PhD scholars normally do, they take different concentrations of crystallizing agents, keep on putting it under different conditions, keep on optimizing the parameters. Okay, then hanging drop method, they would try to fall the solution from a height and try to form the crystal. Still if the crystal does not form, it's troublesome part. Another biggest trouble, see, when the protein is getting apart, expressed, many proteins do not express. They go into inclusion bodies. So you need to express them properly, okay, and then again bring it down into soluble part, and then take the protein from there and then start doing it, forming the crystal. Third problem, see, even if I am able to do it, okay, I got everything done, then the X-ray part I need to start solving it, which takes further more time to analyze all the data. Okay, so it all together includes my X-ray will take around 3 to 5 years, which is a PhD tenure, to solve one structure. Or if I have the data, if I have the protein data, everything, the protein crystal, it will take 1 to 2 months to get the data. Now go to NMR, nuclear magnetic resonance. Here what is the trouble? NMR is applicable, understand that NMR is applicable for the proteins which do not form crystal, so it's okay. X-ray limitation is resolved by NMR. So what is the limitation with the NMR? NMR is applicable for soluble proteins only soluble proteins. So if some protein goes to inclusion bodies, it comes in insoluble fraction. I need to use urea. Okay, I need to use solubilizing agents, refolding kits, protein refolding kits. Do any method, but bring the protein in the soluble form and then work it out. Okay, so these kind of troubles come up. So all these parts require, lastly the trouble is all this X-ray and NMR requires large amount of protein and highly pure protein. Anywhere an impurity come, that will hinder the data. You cannot have multiple similar protein structures. You should have only single protein. No protein isomers also. And no additional additives. Okay. So these are the major limitations. Because of these reasons only, like the number of sequences are high as 200 million, 600,000 compared to just 2 lakh structures. See, substantial gap. Sequences very high, structures very less. Okay, and this gap is constantly increasing. Okay, so we either need very accurate protein structure prediction method or more sophisticated technology to give us the required experimental data in hardly an hour, which is not very close down the line. Okay. So now understand what are the methods for protein structure prediction. We understood that there is a huge requirement of protein structure prediction methods. So what are the methods? The first method is ab-initio de-novo. Okay. This ab-initio de-novo method, what it means is coming from the scratch. Okay, coming from the scratch. What it implies is I know that protein structure, best protein structure exists at the lowest free energy. Okay, so now if I am able to trace the free energy structure from all the possible structures feasible for a protein sequence, I am able to get it. Yes, I am able to get the structure. That is the entire principle involved here in ab initio de novo method. Second is template based modeling. Template based modeling involves using the already known structures, already known experimentally solved structures, whatever are just there. We know that there are 2 lakh structures. So if I am able to use these 2 lakh structures for building the model of a protein sequence, another protein sequence whose structure do not exist, then I am able to build its best possible structure using the known structures. That is the concept of template based modeling. Like I am a like statues are there. So some statues, some molding machines are there. If I am able to put clay inside that molding machine and I am able to impart that structure. So based on that molding part, molding apparatus or molding tool structure, the new shape comes up. Exactly similar is the concept involved in template based modeling. Based on the known structure, the new model takes the structure quite similar to it. Third is threading. Threading means whenever a template based modeling method is not applicable, but I'm able to get small patterns from different structures, multiple structures, and then impart those structure fragments just as a jigsaw puzzle for a new sequence. That is, I am able to take structural segments from several different structures and then I am able to link them together to build a final model. That is termed as threading model. Okay. So now why is ab initio important? I am able to understand that a template based modeling I am threading people are using extensively. So what is the entire need for ab initio modeling? Okay, what is the use? So see, understand that homology modeling tells us nothing about the protein folding and stability. I'll be coming back to this line later on also. Understand that in the template based modeling I'm able to use the protein modeling on already known structure and then build a model. But I've just built a model. I don't know how the protein took the structure like that. But if I'm able to use ab-initio, I'll exactly get the mechanism how this protein sequence took the shape of this structure. Okay. Then some parts which are unable to form, like which you are not able to find in the known structures, or the by using a template based modeling or threading method, any method. So these two methods, whatever, they are not able to make the model. That small local segment I built it through ab initio. So ab initio just act as a supporting tool for template based modeling and threading methods. Okay. And it is the experimental structure determination also. Many times when the scientists determine the protein structure in the lab, some parts are energetically clashing in nature. Okay, means at same position multiple atoms come, which is not feasible. So they change the side chain rotamers. Okay, they adjust the shape of that structure, clashing atom in such a way that the energy minimizes. Okay, we are able to extract the best possible energy without substantially hindering the overall structure. Okay, so it tries to remove the atomic clashes in the generated structures in the lab. Okay, so this ab initio method tells us about the mechanism. It helps the template based modeling and threading methods. It is used to energetically refine the experimentally determined structures. So ab initio is also very useful. So I'll start with the basics. We'll start with the basics. So see the this is exactly there. If I'm able to resolve a protein structure at 0.8 Armstrong in the lab, I'll be able to get hydrogen atoms. Exactly like I'll be able to map hydrogen atoms. But scientists rarely reach this high resolution. Remember the concept I again told you. Resolution means able to discriminate two close atoms present at a distance of zero point 0.8 Armstrong as two different atoms and not just one atom. Okay, this is a covalent bond valency. See, 1 to 1.2 individual atoms. See, 2 to 2.5 I will be able to discriminate the amino acid side chains properly. 3.5 I'll be able to overall trace the polypeptide chain. 4 to 5 Armstrong that resolution I'll be get the protein shape, but can't do further. So I need to go very close, high accuracy up to zero point 0.8 or zero point 0.9 to get the structure the best possible way. So only there's a beamline machine in the France, X-ray structure machine. That determination machine is very accurate. Okay. So see, this is how you view the protein structure. Like PyMol is just one tool. Okay, computer tool which is used to visualize the protein structure. Chimera is there, like Discovery Studio, many such tools are there. You can visualize the protein structure like that. And this is the ribbon conformation. See, I'm showing the helices here. You can color it the way you want. See, this is some different amino acids or different chains which are shown as green here. See, always once the build structures are built or experimentally determined, you can see it using PyMol or Chimera or Discovery Studio, like this kind of different tools. Alright, so now understand how can I use these three methods or where to use what. See, understand that when the sequence, if I have a new sequence, no novel sequence, I want to build its structure. So if that sequence, when I'm checking in BLAST or any similar tool like HSPED, okay. So when I'm comparing it with the known structures present in the database, PDB database, if I'm able to compare my sequence against the known structures, I find that my sequence is having a very low sequence similarity or nothing. Only 5 to 10% similarity, nothing. Means out of 100 amino acids, only 5 to 10 amino acids are found similar. And that too scattered, means I'm unable to get anything. Okay, so I need to use ab-initio modeling. However, if I'm able to get at least 30% similarity, again 30% identity, I'm able to use template based modeling. If it is greater than 10% but less than 30%, I can use threading. Okay, sequence length, as because I'm using ab-initio modeling, I need to build right from scratch. So more is the like a sequence length, more will be the number of possible structures for that sequence, more will be the complicacy. So for this reason, if the sequence length increases higher than the 150 amino acids, the complexity increases because number of structures will accordingly increase exponentially. Okay, but for the template based modeling and threading, as because they are like dependent on the already known structures, for them there is no trouble at all. There is no trouble at all. See, you can whatever be the sequence length, as long as they're able to extract a most similar protein structure from the PDB database, they'll be able to build the structure. Search space. See how many search space means how many possible structures can be there for that sequence. As because ab initio is doing right from scratch, so it's very large, enormous number of sequence structures will be possible. Okay, so search space will be substantially large. It needs to search various structures to get the best structure. But template based modeling and threading, as because they are taking already known structures, they know I need to take from here. So whichever the part is not aligned with the template, whichever the sequence segment which is not found similar to the known structures, only for that part I need to build the structure, and other part I can just link it to build the model. Okay, required computational time is too much because it is extensively processing. Okay, here it is too low. Template modeling is very fast. Threading is like faster than ab initio modeling, but is much slower than the. See, threading is much faster than ab initio modeling, but it is much slower than template based modeling. And template based modeling takes very less computational time just to build one model. One model it will hardly take you like to repeat the complete method hardly a minute or few seconds, depending on the sequence length. So threading will take large time, like ab initio modeling will take very large time. Okay, maybe many days also. Simplicity, ab initio modeling is very complicated, of course, because it involves many equations. Like you need to energetically refine the structures, find out all the possible conformations, then keep on building all, then search the space, search the entire structural space, then get the best possible structure, free of all the clashes. But TBM is like very simple, and threading is less complicated. Okay. So what are the steps you use for? Since several methods, several steps which are used for homology modeling. So see, what I'm doing is now we are slowly extending the discussion onto the homology modeling. Like, I need to search the structure similar to the query sequence. Query sequence means it's my sequence of interest. Okay, and then once I'm able to get this sequence, like get the structure similar to it using BLAST, basic local alignment search tool, or HSPRED, like using a based on hidden Markov model. Okay, so where like why we call it a structure based sequence profile, because I'm having my known sequence which is null sequence. For now this new sequence which I want to build the structure, I'll compare its predicted secondary structure with the known structures present in the database, and then I'll be able to build a profile. So it's extending the sequence information into the structure to properly adjust the gaps coming up in the alignment, to refine this alignment properly, and then derive the best possible structure. So this alignment between the target and the template, or the new sequence and the template, is substantially different than the one generated by the BLAST, which is using only the sequence based gap penalty methods. Then the selecting the template. So see the for selecting the template. So either what is the purpose of predictor model? If I want to use the predictor model only for like drug discovery methods or say like ligand identification, I can use any template, which is very highly similar. But if I am able to get some phylogenetic data also, I want to retain that evolutionary information also, I need to use, I am bound to use like a known structure which is quite comparable to the like close to my sequence in the evolution. Like if multiple structures are very close, properly done, I can take the structure template structure whose E value is lowest, because that is the most accurate protein structure. Okay, so data generated, protein model generated by that structure will be much accurate. Well, align query sequence with the template structure. Of course, I need to align it using the gaps. Then I need to build the model. Okay. Now once the model is built, I need to evaluate it. Evaluation I can do RMSD. Like I can compare my model against the template. How much like my this aligned segments are able to properly fit against the known structure for the part which I have copied. Okay, if that is RMSD between them is very low, means the template structure is exactly mapping here in the model as well. Okay, if some RMSD is coming, means there is like a 5 less than 0.5 to 1 Armstrong. This is only because we need to like incorporate the additional segments also to build the new model, to drive the new model. So it is correct. Okay, if it is unknown, if the answer is unknown, I don't know. Okay. Then I can use Ramachandran plot RMSD against template. I can use it. See? So after refinement, once we are done, we'll call it as near native model. Native model is the best possible structure which you can get through lab. But it is very close to native structure, actual structure. So I call it as near native. See? The answer. What is the answer for the first one? Of course, the answer is all of the above. What is the structure builder? Which structure prediction is more used or mostly used? It is template based modeling. Okay. It's applicable for every protein sequence, right or wrong? I'm sure you will be telling false, because homology modeling method, do you use it? The sequence must have a very high similarity with the known structures, which is not feasible always, which you won't get the same condition always every time. Okay, give application of protein structure details over the sequence information. What is the answer? See, the reason is sequence does not give you the functional inference. Okay, what is the function, how it works, and all the different details. So to get those details, we must like find out the protein structure. Now we should extend our discussion to the ab-initio structure prediction method in more detail. So see, like what the ab initio method basically does is, it applies the Schrodinger's energy equation. It has got different variants. Okay, so don't really get confused with the Schrodinger's energy, but the basic concept is the same. It is having the energy equations which is used to calculate the protein structure energy. Okay, that energy equation may have got multiple terms covalent energy, ionic energy, all these atomic parameters, the parameters which contribute to the overall energy of the protein. Okay, will be calculated. Then the concept is that understand that suppose if I am having a protein structure of 100 amino acids, it means that every amino acid has got 5 psi and omega amino acid, omega angle. Okay, it can have, so for 3 into for 100 amino acids it will be having 3 is to 100 conformations, or in another term, if I am able to represent one amino acid using XYZ coordinate, so for 2 amino acids how much it will take, like 3 is to 2. For 5 amino acids, how many number of coordinates possible will be there? 3 is to 5. Likewise, for 100 amino acids, it will have a 3 is to 100 amino acid conformation. These many possible conformations will be there. So if I calculate these, it will be huge number. So it will be coming up here. See? 10 to 10 to 47. Astronomical number. So now see, if the fastest motion between the atoms take 10 to minus 15 seconds. Okay, so it will take 5 into 10 to 32 seconds. This much time to calculate. See, which will take total second is 3.1563 into 10 to 7. See? A huge time span, which is not feasible. So you understand that this, if I go literally by calculating all the energies and calculating all the motions, then working out, it won't be feasible even for 100 amino acids. Means I need to calculate some approximations, which is exactly what is worked out in current science. Okay, so in the energy questions, whatever is there, we try to optimize our part with 2 steps. First is finding the energy. We'll set an energy equation. Then how to take the next step. Once a model is built, how to take a next step, rather than randomly arbitrarily constructing the next structure. What it does is either it try to finding the randomizing coordinates, which are randomizing the coordinates which are causing the clashes, and then again randomize it. Okay, so always randomization of coordinates which are causing the clashes in the overall model will result into new structure, which will lead to the new structure and then again calculated. This step is constantly being iterated until I reach a saturation energy, energy gets saturated with no substantial deviations. Then I say this is the best possible time to stop, or I set the number of cycles. So one is the energy equation which is there, second is the time, how much steps I should take, how to find out the next energy, next equation, next model. Okay, so these are the 2 critical steps. Okay, so what is the assumption of ab-initio method? The protein folding involves a free energy decrease. Of course, when a protein sequence gets folded in a cell using chaperones, the energy gets decreased, and the protein sequence takes the structure in a fraction of milliseconds, and the native structure has the least free energy, global minimum. Okay, although it may not be true for some cases, but yes, it is mostly there. Okay, there's the conformational sampling, finding the lowest energy one, which is practically impractical, as I just explained you. So you need to energy minimize. calculate the potential energy of the unfolded protein, and introduce small changes in atom locations which is causing the clash, and recalculate the energy. And we need to keep on doing and doing again and again until it reaches a saturation of the saturation curve. Okay. Now check here. Assumptions of the free energy or physical energy approach or ab-initio method. Protein folding involves a free energy decrease. The native structure is the least free energy, global minimum. Now see if I am able to calculate this sequence into the overall structure. Find the lowest energy one, native. If I call that energy of this structure is G2. Okay. Calling it as Gibbs free energy. See. Free energy of here, this structure is G2. Free energy of this structure is G1. Okay. So I will derive a mathematical equation, mathematical description of the total energy of the system in a given configuration is calculated as defined as force field. Okay. So force field definitions keep on changing. Okay. For DNA, protein ligand complex, different force field equations are there, which are just changing either the coefficients involved for the different energetic contributions or different parameters turns up there. Okay, like steric clashes. Okay. If I do G2 minus G1, it will turn up as if this structure has a lower energy than here. The delta energy will be less, delta G will be less than 0. But now understand a part, if I start right from scratch, I don't know the actual structure. This structure is unknown to me. Then I don't know G2. So if I want to start just with G1, I do not know how much lower energy should I go. Understand? So say suppose minus 100 kilojoule is the lowest, which I am assuming for my sequence. That my sequence is not taking below 100 kilojoule. So it may be 100.1, 100.2, 100.7, or 99.3, 99.1, like that. So, but suppose the best structure which I am calculating in the lab turns out to be minus 100.9. So any structure I calculate so far, which I have shown so far, which is not close to minus 100.9, are all close to answer, but not exact answer. Okay. So this method is the major trouble is like how much an amino additional energy should I go? I do everything. I saturate the energy, do everything. But like how much lower should I go? Okay. So if I'm a bit higher or bit lower, then the native energy is here, minus 100.9. If I take it to minus 111, 111 or minus 107, I keep on moving around the native. Okay, but I don't have the actual answer because I don't know. This is exactly the problem which turns up. But still, I try to find out the best possible conformation. And this, so where could be the error which is causing this difference? Only because of like few atoms which are not properly stabilized in my structure, even under best conditions, which get stabilized in the actual answer. Okay, which is still far apart. I'm unable to pack all the atoms properly in their lowest energy conformation. Okay. So these are some complexities of the ab-initio conformation, ab-initio based prediction models. If my starting conformation is here, starting configuration is here, and these are local minimums. This side is energy decreases, then this side increase. This is a local energy value here. Global minimum means lowest energy. This is energy bar. Number of structures are vector space. If I keep on sampling, keep on constructing different protein structures, keep on sampling my initial structure, I'll reach my energy here, global minimum. But normally what happens, I reach, as I shown you, minus 100.7, minus 99.3, I get trapped somewhere close here and never reach here. Although this is my ultimate objective, to reach here. And this is sometimes energy barrier. It's because if ab-initio tries to construct several structures, we try to randomize the atoms which are causing clashes. We always try to escape by taking the step parameter. Okay, because we need to walk around the energetic space. This is a okay, and this is we call as energetic landscape. Energetic landscape, the number of the map of all the energies of all the possible structures for a protein sequence. If you put that in a 3D part, this is termed as three-dimensional energetic landscape for a protein sequence. What it shows you, it shows is like if I'm anywhere on the sequence, anywhere, I need to build a final energy model, which is having the lowest energy in all the entire structures. Million structures or billion structures, this is having the lowest energy. Anywhere I'm there in the map, I need to reach here. Okay, that's the entire part. So it's not so simple here. It is in three-dimensional and it can be in anywhere. So option 1 is sampling all possible conformations and finding the lowest energy one. It's practically impractical as we discussed. Second is energy minimization. Evaluate the potential energy of unfolded proteins, introduce small changes in atom location and recalculate. If energy is lower, accept. Do steps 1 and 2 again. Keep on doing it. Do these steps again. And keep on moving it. Until another, make another change until energy is lower. So after some time, energy gets saturated as we discussed. It does not go beyond. So normally, we try to reach here, but it gets trapped over here. Okay. Hmm? Decrease in free energy. We keep on calculating the new structures always. See the energetic landscape, which I just told you. See? I've taken it from a research article. If I start here, this blue zone is the lowest energy conformation. Here, right here is the global energy minimum. If I start right from here, my initial model is here. I need to travel across all these to come here, which is practically infeasible, no, not feasible at all. See, are you understanding? So, see this is the biggest trouble here. See, like if I see different positions here, they are all different structures. Okay, as we just discussed, energetic landscape means 3D map of all the possible structures, okay, or their energy. Okay, so at every position there's a different energy for a model which is constructed at that place. Okay, as because like some structures have a higher energy, some structures have a lower energy. So at different positions, there's a different energy there. Okay, if you plot them together, you will get a 3D landscape like this. That's exactly the challenge here. See, all these different structures are shown here. See, all of them are shown here. See, this is the native energy, conformational space, and this is the potential energy. You need to go down the hill to build the ultimate lower energy structure. So why is ab initio important over others? As we have been discussing, because ab initio homology modeling tells us nothing about the mechanism of protein folding and its stability. As I earlier told you that I'll explain you this line later also. So now understand that if I build homology model, take a known structure, align it with the unknown sequence, build the model. In that case, I never construct this kind of analysis of 3D landscape, 3D model using the energy, or I minimize the structure like this. I don't have any idea how the ultimate lower energy conformation was formed, or what all possible energies were there within the 3D landscape which is closest to this structure. I don't have any idea. I directly start with a new sequence, align it with the template, build the model. Okay, within a minute, within a fraction of seconds. Okay, but if I do ab-initio modeling, it as it helps fold recognition, there is threading method and homology modeling, refinement of the structure, like build large loops and side chains. Okay, refinement method is same, like you randomize the clashing atoms and again build the model. That's how the refinement is done until the lower energy structure turns up. Large loops means the loops are the structures which are not aligned with the template, like the amino acid regions whose corresponding template information is not existing. So I am bound to model it using ab-initio method, using the basic free energy principles. And then for the side chains, I take the complete structure, keep it intact, and then like keep on putting different side chain automers, and then try to pack the structure to its lowest energy structure. Okay, that's how ab initio methods are used in both, like threading, that is fold recognition, and the homology modeling method, that is template based modeling. Okay, because it can ease experimental structure determination, we are using it for like minimizing the energy of the experimentally determined structure also, to remove its clashes. Okay, it helps to remove atomic clashes in the generated structures using experimental determination as applicable for a protein sequence. It thus finds the reasonably correct model among all structures possible for a protein. The energy map of different models is termed as an energetic landscape, which is just shown here. So this is the optimization problem here. Define some initial model. Where from I should start this? Okay. Yes, because see, if you go here, you get this kind of structure, and it will take a long time to reach here. If you start here, you may build the initial model faster, but we cannot control it. Okay, whatever the structure is formed, okay, as it- we start that whatever the structure is built, like we need to start right from there. And we don't know where exactly it is, because we normally don't build all this until I calculate all the energies. Okay, which will take too much time again. I don't have this idea, but I keep on minimizing the energy with an assumption that this energy will be the lowest and will be the best structure. I keep on following this approach rather than building all this. Okay. So define an energy function to compute and the best model among the numerous strong structures. Okay. You need to be able to figure out what is the best structure. So towards the end of this session, we will be discussing the force field, and then I'll be showing you we'll be discussing a few questions for checking the practical concepts that we have learned in this session. Force field is the energy equation that is used to quantify the potential and energy of a molecular structure. Okay, like protein ligand, including DNA also. So for any new element, also any new complex, any new structure, I also need to calculate the like parameters which are there, like energetic parameters which are there, and then calculate the energy. Okay, it uses the mathematical equations to calculate the potential energy of a molecule, which includes van der Waals forces, covalent bonds, hydrogen bonds. Okay, you understand that hydrogen forms in like a bond with the fluorine, oxygen, nitrogen, as was oxygen and nitrogen are abundantly there in the protein. So these are there. Okay, and then charges, different other kinds of charges. Okay, hydrophobic effects. So considering all of them, the protein structure needs to be computed. The energy needs to be computed. Okay, so towards the end of this session, we'll be evaluating our practical understanding so far using some conceptual questions. So the first one is the protein is modeled using a modeling methodology. Any one of them means ab-initio, template-based modeling or threading method. State under what condition the following observation should be observed. Means if the template is like this, predicted model of protein X is like this. The RMSD between them is 2.9 Armstrong. As we discussed, if the RMSD between a target and a template is nearly between 0.5 to 1 Armstrong, this is very accurately modeled, which could have been possibly for the template-based modeling or threading method, or homology modeling or threading method. But here it is not the case like that. Okay, so what it implies? It implies that it should be possibly homology, this ab initio method. It should be ab initio method. Moreover, if you properly see it here, it is 2.9 Armstrong. If I have used ab initio method, like it could have gone substantially large as well, but still I am finding it 2.9. Means ab initio method has somewhat gone correctly, but not remarkably accurate. Means the sequence length for this protein is less. Okay, because of which it is able to, ab initio is able to properly pack it. Despite the limitations of ab initio method has properly packed the structure to make the model look exactly close to its closest template, like most similar template, like the template and this target is whatever be the case, but ab initio method has resulted into 2.9, means the length is substantially very less, and which can be like difficult to guess from this. Okay, so the predicted model might have been predicted using ab initio method, so only its RMSD is very high. Of course, hence the modeling method might not have properly sampled the conformational space of the target. Yes, that's substantially there. Because unlike 7 to 8 Armstrong, which could have been expected, because it is ab initio method, it is lesser. RMSD is lesser, 2.9 Armstrong. Means the sequence length may be less. But this model sampling might not have been accurately worked out. Okay, you remember we discussed where the clash is there, you need to randomize the coordinates and again construct the model, and you keep on following these steps until you reach a like saturation energy saturation curve. Okay, energy gets saturated. But here it is not like that. Okay, so this has not been the case. Again, sequence identity between the target and template is very low, of course. So only we have taken the ab initio method here. Okay, and also only the model is not exactly like the template, giving high RMSD. That is of course there. Even if it is anything, I cannot use this template for building my predictor model. using ab-initio, because I cannot ab-initio modeling methodology, remember that, is not dependent on the already known structures. I cannot use them. Okay, it results into a high RMSD. However, if the RMSD is lower, then I can say it is built using template based modeling. The alignment has gone properly correct. Okay, there are not much gaps there, because of which the model is properly packed. The first model built using the template is very accurate. So even if I am doing the sampling, protein model sampling means building more and more structures and refining the clashing atoms, energetically placing the clashing atoms to their best possible structure will go correctly, which will further minimize the RMSD between the template and target. Okay, because see, understand that energetic refinement is also termed as energetic relaxation, because I'm relaxing the protein structure to pack all the model atoms properly, okay, to their lowest energy conformation. Okay, and that's the best part. Initial modeling methodology is helpful when sequence similarity of the target protein is very low against the template. This is correct. Target sequence is very large with no significantly scoring template. This is of course correct. Because the sequence similarity is very less against the template, and if the target sequence is very large, not much templates will be able to cover it in a significantly similar way, because where the e-value will decrease. So here the e-value within the target, e-value of the template will be substantially high. So the answer is both A and B. Okay. What is the major problem to use ab-initio method? Its resultant model may not be correct one. Is this correct? It shows numerous structures for protein sequence or chunk of amino acids. That's also conceptually correct. So many structures are feasible. Okay. So many structures are feasible. Among which, if you choose the best model, the selection becomes another criteria, another trouble. Okay. So both A and B becomes the right answer. Okay. So ab initio, let's see the next part. Ab initio method models a sequence on basis of the energy function force field. Yes, correct. To, to only get the lowest energy structure, and then attempts to appropriately move through the energetic landscape of a model to build its final model. True or false? Of course true, because it only has the energy function. Using the energy function only, it is able to build the model. By always refining the atom, You remember the energetic landscape, 3D energetic landscape, where anywhere we are there initially, we need to move closer to the global minima. That's our ultimate objective, which is we are believing that this global minimum is the native structure. Where is the native structure of the protein? That's an assumption. Because protein structure exists at the lowest energy. That's our assumption. Based on this assumption only, it is using the physical principles, that's the energy equation and the step parameter to move over the energetic landscape to build its protein structure. So it is true. Can there be any method other than the energy to evaluate these protein models? What you will choose? I need to choose the models, best model. I am moving through the energy, correct. But other than the energy which is used for movement across this energetic landscape, can there be any other method possible to assess the best model, that this is the best model besides energy? Yes, I am sure you will be thinking one term or few terms. Like the most simplest of which at your level is like proper packing of the protein. So the protein structure is completely packed. Okay, that will be the like accurate structure. Like protein structure should not be like opened up. It should be properly packed. So how will you calculate it? You can find out the length of all the atoms, diameter of all the atoms, like the highest diameter feasible from end to end, and then when you sum them together, it should be there close to the like overall diameter which is there. So if the overall diameter between the end to end diameter possible for a protein structure is lowest, then that is correct, not like opened up some vague structure. So we have reached the end of this session 3. I hope you certainly might have enjoyed it. Goodbye.